Okay, good evening, everybody. Welcome to Cabri Aquarium's Discovery Lecture Series. I'm Julie Passarelli. I'm the Exhibits and Collections Curator at Cabri Aquarium. Cabri Aquarium is owned and operated by the City of Los Angeles Department of Recreation and Parks, and we're extremely grateful for the City of Los Angeles' support. I would also like to thank the Aquarium Director, Chrislyn McCarran, and Jim DePompe, the Programs Director, for all their support, and a huge thank you to everyone who's joined us tonight. The aquarium remains closed due to COVID-19, but please check back on our website for any new information about opening dates and some of our online programs. Before we get started, I would also like to thank and acknowledge the Friends of Cabri Aquarium for all their support. We also plan to continue the lecture series through the rest of the year online. The upcoming lectures are November 6th, also a Friday, November 6th, Dr. Nicole Benuso from Cal State University Fullerton. She'll be talking about bringing dark data into the light, one shell at a time. And then the last lecture of the year is also Friday on December 4th. And it's Dr. Jay Smith from Cal Poly Pomona, and he'll be talking about rocky intertidal ecosystems in urbanized Southern California, long-term change, human impacts, and management. If you're interested in any of these lectures, please mark your calendars and check our website for details and to RSVP to receive the webinar link just like tonight's. So it's my pleasure and honor to introduce tonight's speaker, Dr. Nina Bednarsek. Dr. Bednarsek is currently a researcher at SCCWRP, the Southern California Coastal Research Project. She received her PhD in biologic oceanography from Cambridge and started her research a decade ago in Antarctica. The polar regions have remained her passion ever since. She is committed to communicating research to various different audiences, from schools, NGOs, to governments, to international policymakers. Her research focuses on the understanding of biological effects and the risks of climate change in marine organisms. She specializes in determining the tipping points related to ocean acidification in combination with other stressors on the most vulnerable calcifying species. The title of tonight's talk is "The Risks of Climate Change on the Most Vulnerable Species and Ecosystems, from the estuaries to the coast, all the way to the poles." Please help me welcome Dr. Nina Bednarsek.
Okay, so thank you so much for the invitation, and it's my pleasure to sharing some of this science with you. And so tonight, if I can start this, it's actually all about the oceans and marine life and how we are changing it and how it's changing under the climate change. So I want to take you to this journey across different ocean regions and show you some examples on how humans are changing the ocean habitats. And in this talk, you're going to hear a lot of negative facts, but I want you to remember that all the changes we are seeing, they could be stopped if we collectively as humans can start taking actions right now and try to reverse the process. So let's start with some facts that IPCC, which is the Intergovernmental Panel for Climate Change, has recently published. So IPCC comprises a wide panel of top scientists across the globe, and they have recently produced this special report on the ocean and cryosphere, in which they basically synthesize all available science data and they bring new conclusion on the state of the ocean. And what they're basically saying is that the oceans are projected to transition to unprecedented conditions. So something that we have not seen any time before in Earth's history. And the thing is that the oceans are really changing in multiple different ways. They are warming up, not just in the surface that is exposed to the warm air, but also in the bottom because they're absorbing all that excess heat. They're losing breath. This means they're containing much less oxygen than they did in the pre-industrial times. And they're turning sour, so they are becoming more acidic, more corrosive. And in addition, they're experiencing much more of the extreme events, like marine heat waves, as we call them. And if you remember, in the period from 2013 to 2016, we had this phenomena called the blob that completely reconstructed North Pacific biological community to the extent where we only start to understand it now. So all these impacts are happening in the oceans, and I want you to notice here is how different the oceans were in the pre-industrial times, and that's the brown lines that we have indicated here, compared to the trajectories that they're on now. And these trajectories we can delineate with the RCP scenarios, and RCP stands for representative carbon pathways. We can have two scenarios, a lower emission scenario indicated with RCP 2.6 and higher emission scenarios standing with RCP 8.5. That's a business scenario it's a scenario as business as usual where we are not slashing any of the emissions. And all the figures, it's basically obvious that the negative impacts on the oceans are so much more detrimental under these RCP 8.5 scenarios compared to 2.6. And it's really is the change in the scenarios that would either sink or save our oceans. All right, so where is all this anthropogenic CO2 coming from? The majority is coming from the combustion, so burning fossil fuels, transportation, cement production, and tiny bit is coming from deforestation, so losing the forest, cutting down the trees like it's happening in the Amazon. And so with all this CO2 in the atmosphere, approximately 50% is staying as airborne fraction, but the other 50%, half of it is absorbed by land, and the other quarter is basically absorbed in the ocean. And this uptake of CO2 in the ocean is actually very well understood phenomena. So we understand the physical chemical processes, the thermodynamics, and the impact of ocean circulation on how it impacts the ocean. So when you hear the criticism that we don't actually understand these processes, how the CO2 from the atmosphere travels into the ocean, that's not true. We have a very good handle on that. So I think I really want to stress out for you how important this uptake of CO2 in the ocean is, because it really buffers us, protects us from the worst impacts of the climate. Had it not been for the uptake of CO2 in the oceans, all that CO2 would basically stay in the atmosphere and cause much more serious climate change effects, as we are already seeing them, from extreme events to hurricanes, increased sea level rise, and so on. So ocean is really helping us in buffering a lot of these effects. But as said, climate change is still a triple threat for the oceans, right? Getting warm, more acidic, containing less oxygen. I mean, that is definitely ocean habitat that is not, favorable for marine species. And a lot of different things is happening that are detrimental for marine ecosystems. We're seeing increased sea level rise, and that's going to have implications for the coastal communities, for coastal erosions, and so on. The corals are bleaching like never before, and it's really difficult to reverse those processes. We are also seeing an increased toxic algae frequency in our warmer oceans, and we're continuously degrading the habitats with less oxygen that some species can no longer inhabit. When it comes to ocean acidification, we are basically making the oceans more acidic, and that prevents some of the corals, clams, and oysters more difficult to build their shells, but it also has an impact on the fisheries communities. It will endanger the global food security and the global food webs. So there are a lot of implications on how climate change can impact the ocean, and we could be here forever if we wanted to tackle all of this. So in my talk, I just wanted to focus on this ocean acidification and tell you more in detail about this process, so we can really have a focused look on one of these processes here. Okay, so what is the ocean acidification? This is a 101 lecture in here, really. So when carbon dioxide from the atmosphere comes and absorbs in the ocean, and you have to know that whatever it's in the atmosphere eventually gets up in the oceans, because those two are in equilibrium. So CO2 binds with water and starts forming carbonic acid. but that acid is not stable. So it very quickly dissociates into the hydrogen ions, so protons, and this makes the oceans more acidic because it lowers the pH. But it also happens so that we start losing these carbonate ions, and carbonate ions are really important for shell-forming species to form their structures. So with that, we have oceans that are much more corrosive, and the shell-forming organisms cannot really function in there. And I just wanted to show you that the ocean acidification has multiple biological effects, from these increases in CO2 and pH and corrosivity, and all that can impact biological communities. So the carbon changes that we know of are happening already. So this is not the case of future scenarios. If you're sometimes hearing, okay, so this is going to be the world by this and this year. In fact, the changes with ocean acidification are already here. And so the upper graph in here shows surface water CO2 that is basically increasing at the same rate as the atmospheric CO2 emissions, right? As I was telling you before about the equilibrium, so whatever we are putting out is going to get absorbed in the ocean. So this is the data from Hawaii Ocean Time Series. It's an isolated place in Station Aloha, and really whatever is happening in the oceans, we can have a good handle of these processes in there. And with the increases in the CO2 in the water, we're basically declining the pH, which is the measure for the acidity level as well. So be aware that oceans are not acidic, but with increase of these protons, they are becoming more acidic. So all this is already measurable. We have instruments that can show us what's going on in the water column. And so we have a good understanding of what that is. But one thing that we are extremely concerned about is how quickly the oceans are changing. So the rates of increase, and here I'm showing the atmospheric CO2 in blue, and then the global temperature in red in here for the last 100 years and the next 100, and in two different scenarios, 2.6 and 8.5, so lower versus higher emission scenarios. And basically what we know is that if we follow the RCP 8.5 scenario, so the emission scenarios in which we are not slashing our emissions, the rate of change is 100% faster than anything that has occurred in the last 100,000 years in Earth's history. So we have never seen such changes before. And that is really a primary reason for our concern, because we know that the oceans cannot adapt so quickly. They don't have buffering capacity for such high increase of change, but also the organisms and the ecosystems that are inhabiting the oceans have no adaptation to such high rates of change. So it's not only the magnitude of the change, it's also how quickly we are introducing these changes in our oceans. So when it comes to ocean acidification, now we know how we are changing the chemistry, but what really matters is how the biology changes and how that's going to impact the humans, right? Because if we're affecting ecosystems, we are directly impacting people and their resources, food webs, and so on. We also know that the biology has already changed quite a lot and is going to continue changing under future scenarios. And that's going to have social and economic repercussions. So either we're really changing the food webs, the fisheries, and so on, or we're going to destroy or significantly reduce our ecosystem-related services. And that means money, that means resources for humans that are directly impacted by these changes. And usually these are more vulnerable, poor communities that are directly dependent on the oceans. Okay, so all this actually calls for us accurate understanding and predictability of the current and near future biological responses, but also solutions, right? So, I mean, one thing is just to observe the phenomenon. The other one is trying to put the solutions together and try to save what we can save in this amount of time. So we are talking about both the mitigation and adaptation to climate change in order to save the oceans. Okay, so this was a little bit of a global perspective. Let's go to back to home to our California current system that is also changing with very fast rates. And I just wanted to show you some of the background of the processes that we have in here so you can understand why the California current system is specifically vulnerable to ocean acidification. So along the US West Coast, we have these natural processes that can accelerate ocean acidification in the coast. And the process is the following. As we have the wind stress coming along the shore, that basically pushes the coastal waters offshore and replaces it with the deep waters coming through the coast. Now that deep water is really high in CO2, it's very corrosive, and it's actually acidic. And all that is coming into the surface. That process is called the coastal upwelling. Now, this is a little bit of a different representation of that deep water coming into on the continental shelf. And what's going on is that deep corrosive waters is then exposing these productive coastal ecosystems to this upwelled water. So this is a natural process, but now we are changing the strength of the upwelling, and all the CO2 that is coming from the atmosphere into the ocean that is changing because of the climate change, that can significantly basically increase the strength of the upwelling. So much more of the corrosive water is now coming onto the continental shelf. And as a result, we have actually expanded the ocean acidification along the west coast. This is a study that we have done in 2014, and it's basically comparing how the west coast and the ocean, the coastal conditions along the west coast were in the pre-industrial times. That's the panel on the left. In 2011, that's the panel in the middle, and 2050, the panel on the right. And so what I'm showing here is the percent of corrosive waters in the upper 100 meters. That is the important pelagic habitat for a variety of different shell-forming organisms. And the color bar represents favorable versus less favorable conditions for these biological organisms. So favorable darker colors and not favorable with brighter colors. And if you see in the pre-industrial times, most of the US West Coast was favorable in their environment. Just Washington and Oregon area had some of these corrosive conditions. But by contrast, in 2011, we have seriously expanded these corrosive waters in Washington and Oregon areas. But if we now see what happens by 2050, we've estimated that approximately 70% of all the waters get corrosive. You see now it's not only in the coast, but it's getting offshore as well. And so we are basically significantly reducing the habitat that shell-forming organisms can inhabit. So it's a huge and rapid loss of a habitat and marine ecosystems. So what sort of effects on the marine organisms would such expansion of ocean acidification have? We're predominantly concerned for marine organisms, for marine calcifiers, so from bivalves, sea urchins, corals, crabs, all the aquaculture important species, but also fish as well. And I showed you before how it's not just pH, so it's not just ocean getting more acidic, it's also getting more corrosive and losing these different parameters that is really important for a variety of different organisms. And these are just some of the biological impacts that we're gonna see. So when this changes, we're gonna start seeing reduced calcification. So organisms having problems building their shells, they will not be able to grow, but some of their shells will also be dissolving. Especially for the fish, they have observed that CO2 can cause their changes in behavioral responses. So fish no longer swims away from the predator, but towards it. So really fundamental shifts in neurological responses that can of course lead to the changes in fitness and survival. And on a larger scales, we're gonna see the changes in the species biogeography, shifts in diversity. Now, you can only imagine that if you are impacted by ocean acidification and you're compromised by that stressor, then you will have much lower tolerance to the other environmental stressors. Like if you are hit simultaneously by warm water and water that is low in oxygen, you'll have much more difficult times if you're already living in the corrosive conditions already. Ultimately, that will shift the food webs and be a really important factor on declining ecosystem goods and services. So this is this multiple tiers how we're going to start seeing, or we are already seeing these negative effects in the oceans. Okay, so what have we learned so far? So let's dip into the science a little bit more along this open ocean to coastal interface in the California current system, which is basically this coast within the Northeast Pacific Ocean. And I want to show you a variety of different approaches, how we study biological responses, but also focus on two specific groups because they're either ecologically or economically important. And so you can understand how we have actually changed the ocean so far. Okay, so this is our work on board the ship once we go on the cruises. This is a NOAA ship that we have done several different cruises with. And so we are simultaneously collecting waters. The picture on the right is actually releasing the CTD bottles, and they're opening at different depths. So we can actually collect the water from different depths and then characterize the chemical conditions of the water. On the right is basically a picture of biological sampling. With red arrow, you see the bongo net. It's a net that we can collect this variety of different marine invertebrates living in the open ocean. And so this is more ideal scenario. It's taken during the daytime, and that's perfect. But usually that's happening during the night. And, you know, people are usually very tired during the night shift and usually seasick as well. That's why my glorious face on this image as well. Not very happy. But so this is how we are collecting our organisms. It's not all that bad because we have opportunity to set up our labs on the ship. There are specific rooms on the ship where we can basically set up the microscopes, variety of different techniques with which we can measure biological responses on board. And so you see, we have a lot of fun doing it, right? Although it's exhausting and we are really dependent on our colleagues on the ship, it is really a one-shot opportunity because when we go out, these cruises are really long. We go usually from Mexico to British Columbia. So we go this zigzag route over the entire West Coast. And so we're really trying to understand these marine habitats and how much they have changed or what's going on. So trying to establish the status of change in the oceans. So what have we learned about these biological impacts in this open ocean? Okay, first let me introduce the pteropods to you. I'm not sure how many of you knows this species already, but 10 years ago when I started this were new creatures to me, and I was completely mesmerized by their aesthetics because they have this really beautiful thin transparent shells. But what pteropods really are, they are actually snails, where their food has been transformed into two wings. And so by flapping two wings, they can move up and down the water column. So a pelagic snail with a shell surrounded it as a protection. And so they are very small, usually 1 to 2 millimeters, and belong to the zooplankton. Their shell, which is as thin as our hair, is made of aragonite. Aragonite is a crystalline form of calcium carbonate that is more soluble. And they are not present just along the US West Coast, they are basically in all global oceans. But in our oceans, they can be actually quite abundant. And they are, when there are a lot of phytoplankton on which they're feeding on, they can ferociously graze on them. And that makes them an important player of transferring energy to the upper trophic levels. Okay, so and these are just some of the species that are feeding on pteropods. So from macrozooplankton to fish, birds, whales, and although they eat pteropods in different proportions depending where they are, some of these species are really hugely dependent on pteropods, especially the baby salmon, pink salmon, and sockeye salmon are one of the few that their stomachs were absolutely full with pteropods. So an important component of the community in here. But there is a darker side that pteropods represent, and they are the canary in the coal mine for that specific reason. So we knew now for a while the pteropods are one of the first species to be severely affected by ocean acidification, and that's especially because of that extreme sensitivity of their shell, and it starts dissolving under more corrosive conditions. So here we have two images. On the left, it's basically intact pteropods, really nice, smooth shell. And on the left is the pteropod that has been exposed to more corrosive conditions. And you can see it's been compromised. It's much more fragile, and it has a lot of dissolution in here. Are the results of the corrosive pteropods from the Southern Ocean. And this is how pteropods look like in the natural environment now. So I started doing my research on pteropods in the Southern Ocean around Antarctica. And we came and published this study in 2012, where we are first disclosing this evidence of severe dissolution in the natural environment. But the really surprising thing was that if it wasn't for the addition of the carbon from the humans, we wouldn't have seen such severe dissolution effects on pteropods. It was really that added element of carbon coming from the humans that is making the pteropods so dissolved. And that was totally unexpected, because according to all the models by then, we knew that we're going to see these changes maybe by 2050 or 2100. And seeing it in the beginning of the century was really hugely surprising. And it just helped us understand how rapidly the changes in these polar habitats are taking place. But we also decided that this kind of shell dissolution, we can use it along the US West Coast in our regions to track how much pteropods have changed from the past, the present, and how they're going to look in the future. So these three images are corresponding to the carbonate chemistry condition map that I showed you before. So in the pre-industrial time, so that's a time where the humans didn't emit the CO2, and so the oceans were not impacted by human emissions, we had this really transparent, smooth, healthy pteropods. But currently, approximately 10% of all pteropods is affected by some moderate to severe level of dissolution, and that's especially in the coastal oceans. And by 2050, when we destroy approximately 70% of their habitats, we'll also have approximately 70% of pteropods severely affected by this shell dissolution. I mean, in this particular case, this is experimental pteropod that we have exposed to the conditions of the future, but you can start seeing the holes on the shell and severely compromised shell. So, you know, this shell dissolution really has an impact on pteropods and how it can swim. So here is a healthy pteropod that has not been compromised by shell dissolution. And you can see how quickly, how agile the organism is, right? We can even see like the heartbeat of the organism and how quickly it's moving, basically very fast response time to stimulus from the external environment. But on the other hand, this is a pteropod that has been massively impacted by shell dissolution. You can see the holes in there, and the shell becomes really opaque and non-transparent, and it's hardly flapping its wings. It has absolutely no energy to move anywhere. So suddenly, if you can imagine, I mean, swimming is a major tool of pteropods to be able to thrive in the oceans, yet they cannot move. So that alone can tell you how massively they are compromised by these changes. Okay, so here a little bit of how modeling can help us in understanding pteropods, and I think this is really fascinating how the biology and the modeling can come together. When we are trying to understand how pteropods are exposed to ocean acidification along the US West Coast, we definitely have to study how they're moving in these habitats, right? So we're trying to use the modeling and track pteropods as particles there, right? So you deploy the particles in the model and suddenly you have understanding where they go. So we are tracking basically pteropod dynamics in this coastal region of the oceans. And as you can see, all the pteropods are actually coming from the offshore, and they're slowly coming onshore. And in a matter of a couple of months, they can traverse these huge distances from British Columbia to Oregon, and they have high level of spatial connectivity. So we say that is a pan-mictic population, and there are no biogeographical barriers in the ocean that would prevent mixing of these pteropods together. But as you can see, when they're coming from the offshore, more suitable habitats, as indicated by the color red of the particle, they slowly come into these coastal areas and they start getting into these more corrosive waters, but they get retained by the advection mechanisms in the coastal habitats. And this is where the negative impacts actually really start. So this is how we can actually much better understand what's going to be happening with the future populations when we start using such fantastic modeling efforts as here. Okay, switching to another organism, I told you now something about the pteropods, the zooplankton, and now to Dungeness crab, this really iconic species along the west coast, mostly because it's so economically important fishery with more than $300 million revenue per year. And it's both commercial and recreational fishery, also very important cultural and spiritual resource for the native communities. So they have a lot of festivals in which they are preparing this delicious food. Dungeness crab are actually present from California. They're usually in the northern California down to central California and all the way to Alaska. But here we're thinking of the impacts of them and the potential economic impacts that will have for the communities and for the revenues by itself. So pteropods were more ecologically important, but here we have to think about very different dimensions, and that is how the human perspective will be impacted by the Dungeness crab decline. Okay, so in order to understand how the entire population of Dungeness crab is impacted by ocean acidification, we need to understand their life history. So what sort of stages they are going through in their life cycle. So when you see a big Dungeness crab, that is a 3 to 4 year old crab, and has gone through a mixture of pelagic and benthic stages. So adults basically release the eggs, they go to the larva forming, and that larvae, once it matures, it actually settles on the bottom, becomes a juvenile, and from then onwards it lives only at the bottom of the ocean. But we know that in this process, maybe the adults are not so vulnerable to ocean acidification, but the larvae are, and there are two reasons for that. Their exoskeleton of the larvae, or you can call it shell or carapace, is made of the calcite. And so this crystalline form is prone to dissolution. So if they're going to see the corrosive waters, we can expect that they will basically have some sort of level of dissolution on their shell as well. The other important thing is that they have these mechanoreceptors on their carapace, and they're kind of like antenna for detecting environmental cues, right? So they're really important for a variety of different biological processes like secretion, feeding, moving in the water column. When they have to recognize where they're going to go and where they're going to settle, they're using this antenna for detecting the proper habitat that they want to settle down. Okay, and so the larva crabs are actually already exposed to OA conditions because they are vertical. That means that during the night they go to the surface to feed, and then during the day go to the bottom of the water. And so when they're at the bottom of the water, they're exposed to these more corrosive conditions in these nearshore habitats that you can see indicated red in here. And so we've changed the nearshore conditions and habitats to the extent where these larval stages can already be impacted. And so nobody has really thought about this before, and then we start thinking, but surely they are these larval stages. They build their shell out of this more soluble form of carbonates. Let's explore what's happening with these larval tiny shells. You could see like an organism like this, maybe 5 millimeter in diameter. And when we put them under the scanning electron microscope, so this is a microscope when we can really observe their shell surfaces, we start seeing this really strange folds and ridges and a lot of crystals exposed. And so basically they were dissolving externally, they were dissolving internally, and so a lot of changes that have never been described before we have seen on these crabs in this nearshore region. So another similar kind of effect that we have seen previously in pteropods was also visible in these crabs. But what we have noticed in crabs and what we haven't seen in in pteropods is this collateral damage that they have as well. So I was telling you that they have this antenna, and on image A, you can see these spikes coming from their claws, and these are these antenna or mechanoreceptors that are basically anchored in these canals on their surface. And what we noticed is that suddenly these mechanoreceptors were no longer there. And we were trying to figure out the mechanism for there. And higher magnifications have actually told us that the surface around this area starts dissolving. So they no longer can be rooted in the shell surfaces. So suddenly these mechanoreceptors, they destabilize and they collapse. So what is the functional implication for the organisms for that? We have no idea, but that is definitely a novel, another additional pathway of sensitivity for these larval crabs, and we need to definitely study more what sort of implications that has for the organisms. Okay, so here, another thing is, so Dungeness crabs are also present in the nearshore environment, also in the Pacific Northwest, in this big estuary of the Salish Sea. And although we haven't done observational work in there, we have been playing with some of the modeling there, because Washington Department of Ecology has this really nice Salish Sea model. So we were trying to delineate different model scenarios, how these crabs are impacted, right? So depending on the biogeochemical model that you have, you can actually run different scenarios to figure out what is the driver behind the biological impacts. So in this particular case, we were asking the question, is it more the atmospheric CO2 uptake, so basically climate change, or is it more the nutrients that the people are releasing into the ocean that are causing potential negative effects in the Salish Sea? And I haven't told you that before, but when nutrients come into the ocean, they start getting degraded, and that can increase the ocean acidification as well. But why we study the nutrients is because that's a management strategy that people can actually handle. So we could reduce the amount of nutrients coming into the ocean, while we actually cannot reduce the CO2 uptake, because that's a global phenomenon. So we wanted to know, basically with this model, if by reducing the nutrients, we could actually reduce the negative impact on the crabs in the Salish Sea as well. And so this was a very nice study, because it showed us that nutrients actually contributed only 3%, and the rest, all the other changes that we would see in the dissolution in the crabs in the Salish Sea, are mostly coming from the CO2 uptake. So this is a clear-cut climate change effects on the crabs in this big, very productive estuary of the Salish Sea. But why this is so important is because parts of this Puget Sound, or southern part of the Salish Sea, they have been closed to Dungeness crab fishing because of the collapse of the population. And this Dungeness crab in the Puget Sound is really important source for the native communities in there. And there was no understanding of what is causing this collapse of the population. So we now think that by showing how much the changes due to the climate change have been in this region, and how much they can impact the dissolution, that the ocean acidification is definitely the parameter that needs to be considered in the Salish Sea on how it can impact these populations. So anyway, wanting to show you how modeling can help us understand some really important decision-making processes. In this particular case, for example, shall we reduce the amount of nutrients that are coming in this basin or not? And understand that reducing the nutrients is $1 billion industry, and it would really cost us a lot. So that's why running these kind of models and having that kind of understanding is really important. Okay, so what's next from the Dungeness? We only started to understand what is going on with these crabs, right? You know, this dissolution that we are seeing on the crabs, this is kind of like an early warning signal that helps us understand what's going to be happening in the crabs in the long term, right? So they're definitely not resilient to these changes in the ocean. But we don't have yet the idea of how that's going to impact the population, the industry, the crabbers, the people that are immediately dependent on this species for their revenues and for their subsistence. But in studying this, and when we published our study in 2020, so earlier this year, we got a lot of press inquiries, and one of them came from CNN. And after that, they wrote a story that is so out of line, because they're basically saying the Pacific Ocean is already so acidic that it's dissolving Dungeness crab shell, and that it's very, very inaccurate description of what the actual study was. And in this particular case, the media really hyperinflated our story and obscure scientific findings. But what was more worrisome is how it sends this conflicting message to the crabbing community, who suddenly starts calling, hey, is this a real concern right now or in the 10 years time? But also to the people who are maybe indirectly related to this problem. So just a caution here, how media sometimes misinterprets the scientific findings a lot, and how critical thinking and approach to science is really crucial in conveying the message to the communities that need to hear that message. All right, so thinking about the future biological impacts from the CO2, this was a very nice study done in 2015. And it shows basically how different organisms and the ecosystems would be affected under two different, so lower 2.6 or high emission scenarios by 2100. And really, it's in the middle, you have all these species from seagrass, mangroves, pteropods, bivalves, krill, fish, and so on. And the below is the risk of impact and the management options that we have. And basically, this study very clearly shows that under the RCP 2.6 scenarios, we will still have some of the species that might be more vulnerable. So it's not that we're not affecting the oceans by, but by our already existing activities. But in comparison with the 8.5 scenarios, where the risks is very, very high, but more importantly, the efficient management options are really low as well. So if you see the management options on the left under 2.6, we have options to adapt, protect, repair. But if we are running the high emission scenarios and we are not doing anything to slash the carbon emissions, then the management to basically protect and manage our marine ecosystems and species mostly goes away. So that, of course, is a big call for what do we need to do in the oceans in order to try to save them, right? And so knowing that mitigation of our emissions will not come in time to actually solve these marine ecosystems. So what sort of marine geoengineering solutions do we have to think about in order to basically be able to save our options? And these are some of the carbon dioxide removal strategies, or CDRs as we call them. So we can either remove the CO2 from the atmosphere, we can remove it from the ocean with different techniques, we can try to neutralize the ocean, the ocean's acidity, and so on, right? So a variety of different options. And it is true that once we have a good handle of that, we can really do and protect the oceans much more to what would otherwise the scenario be. But it's important to understand, when we are talking about geoengineering solutions, all this is very, very preliminary. So none of this has been done on a large scale basis. This is all in the form of the preliminary testing. There are a lot of gaps that need to be closed, specifically related to detection and attribution. That means, is a specific technique actually efficient with providing a solution or not, right? The other problem is, for example, with upscaling, when we have the pilot that is going on, that is basically working, but we don't know how that's gonna work out on a larger scale. And of course, the side effect, whatever we are putting in the ocean might be having some side effects on the other organisms as well. Regardless of all this, and the CDR techniques are now really becoming popular, and governments are recognizing the need to test and to implement them in the future. But if we wanted to keep our ocean safe, we will need this coordinated network of multiple marine geoengineering activities. So it's more than time that we start working on a variety of these techniques as a scientific community, but also supported by the governments. Okay. And then I think this is a really nice slide because it baffles people. If we are, how are all these changes feasible, right? And a lot of times you hear the argument, oh, we're not yet there. We cannot really do anything against the climate change. But, you know, and this was a study done by the IPCC, so highly credible scientific body. So when they were trying to see, is there a geophysical feasibility? Yes, there are options for that, right? Environmental feasibility as well. Technologically feasibility, definitely going there with a lot of new methodologies and technologies being developed. And economic feasibility is actually in principle also there just has to be an incentive for this to come to the scientific community. But overall, it really is about us. Are we willing to make that next steps in order to start to work on the climate change and indirectly or directly reducing the impact on the oceans as well? Okay, so in my conclusion, I hope I convinced you that whatever we have changed the oceans, this process is already impacting and the marine organisms and will continue to do so. And most of the changes are really because of the atmospheric CO2 coming into the oceans and not because of some other drivers. So we have to start acting and saving the oceans now before it's too late, because once it becomes too late, then we cannot reverse the process. Some of the changes will be irreversible and we will just have to live with that damage in the oceans. So this was more on a scientific talk, and I just wanted to take a few minutes as well to tell you how the science can really help in the outreach and education as well. And I find this very cool example of how the science and art can work together. So this is from a few years ago when I was working with this artist, David Eisenhower from Seattle area. And we were trying to, you know, incentivize this problem of oceanification and how the communities and people can understand it, right? What do you put in the museum in order to basically depict the oceanification and how dire the consequences would be? So we were talking a lot, brainstorming very different ideas, right? Obviously, you have to have environmentally very conscious artists, and this is an iterative process. But the outcome was really fascinating because we had an exhibition, and this was really art and science working together. And it says dialogue with nature in here. And this artist has been basically making these huge sculptures of pteropods. And this is how they look like, right? So this is the artistic expression of the scientific facts, actually. The sculptor called this shell game. And so he had depicted these pteropods and then the solution on their shells. And he put it in this really distinct spiral form that can, you know, talk about life, longevity. But on this down image in the lower left corner, he had packed little kids inside of that, right? So it's our future generations that will carry the impact of our negative the behavioral patterns right now and inability to get ourselves together to start mitigating against climate change. And so that actually a really strong message. But then moreover, he put some of these pteropods also in the coal and showed this dissolution as revealed by the acid wash. So really direct implication of what is going on in the ocean, right? And so the art really is this impacting people on a very different way, more subconscious way, right? And that requires a very different response than when you hit somebody directly with the scientific facts. And so it's maybe more difficult to understand. And the artist says this is basically dancing around the question to solicit the subconsciousness for the answer, right? And so I find that very fascinating and very positive response from people on this art exhibition in here. So with that, I want to finish. I'm really looking forward to any of your questions in here, but if we don't have time for the questions, then please, you can contact me on this various email or my Twitter, and I can help you with addressing your questions further. I just want to acknowledge that I always said that is kind of like a my science, but without many, many collaborators and friends, none of this would be happening. And of course, a lot of the funding bodies here displayed as well. So thank you so much for your attention.
Awesome. Thank you, Nina. That was an amazing talk. There's absolutely time for questions. And there's been a few that have been coming in. So let me just ask a few directly to you. So, okay, from Kaylin, she is a biology graduate student at Cal State Long Beach. And she says that you mentioned how ocean acidification often inhibits growth. And she's wondering if this trend in growth is consistent across all species studied, or if there are intermediate levels of ocean acidification that cause mixed responses in growth, even potential increases in growth among certain species.
Yeah, so I mean, the species that we study, right, have shown these trends, and that's why I was referring to this as a pattern that we have observed. But she's right that there are species that are responding differently to these trends, and some of them are also positively responding to increase in the change, for example. And so we call them OA, ocean acidification winners, as opposed to losers. So not all the species will be equally impacted. So there will be winners, there will be losers. And there are different ranges of concentration that the species will be impacted by. So it's a variety of different responses. And that's why we're gonna see the shift in the communities, because some will not survive, some will survive much better, but usually the trend is towards the diversity getting much lower, so we'll have much less species compared to the previous one, but there will be some of the winners that we will retain in that process.
Okay, another question, thanks Dina. Another question is, from Evie. When you were talking a lot about nutrients. She wants to know, what kind of nutrients are you talking about?
Well, I mean, that's mostly related to the sewage and wastewater, wastewater that comes from the treatments into the ocean, right? That water still contains the nutrients, predominantly the nitrates and the phosphates that can cause the blooms. The phytoplankton can basically utilize them, and then the phytoplankton and being fed on the zooplankton then causes the so-called remineralization or utilization of these nutrients, and in the process of that, they're basically emitting the CO2. So the more nutrients you're releasing into the ocean, although could be a pre-treated water, they would still be additionally contributing to the process of ocean acidification. So it's something that, you know, in very heavily urbanized areas like in LA or Seattle, a lot of this wastewater goes to the ocean, and it can creates the plumes of water that are more corrosive just because the nutrients have been mineralized in those coastal areas and can cause more acidic conditions locally.
Right. Okay, another question from Kim. She wants to know, have you heard anything about how algae that contain calcium, like coralline algae, can they be affected by this and by ocean acidification?
Yeah, coralline algae are one of the most susceptible organisms to ocean acidification. They will definitely see uniformity in responses in different studies. They are growing less, they start dissolving. So are one of those, you know, primary producers or the plants which you would expect that would thrive under the increased CO2, but in fact, because of their heavily calcified bodies, they're much more negatively impacted by OA. Yeah.
Okay. Stephen wants to know, what can we do individually to prevent these negative impacts?
Yeah, and I think some years ago we were talking a lot about what the individual can do for that, right? And so we started to recycling, and some people, some really conscious people never, you know, venture into the airplanes anymore, because that's obviously way too much of a carbon footprint, and carpooling and all that. And I would say that, you know, while all these activities can contribute to a very small extent, it could make people feel happier that they're doing something for the environment. But really, without massive actions, new technologies, and serious mitigation and removal of the CO2, what has already been released in the atmosphere, we will never get there on the individual level. So you know, I don't want to be politically engaged in here, but the only way how we're going to ever do that is by voting for, you know, your constituency that promotes and is willing to do something for the climate change. And we're talking about the massive change that is needed for that. So it's a perfect timing for that.
I agree. Me too. And I was about to say, vote, people, vote, if you're old enough to vote. Speaking of being old enough, there's a question from a 9th grader named Lavanya. She's a 9th grader and she's doing a science research project at school, and I've chosen to do a project on ocean acidification. Is there an email way to contact you in the future? Is it right if we put your email in the chat, or?
Yes, for sure.
Yeah, okay.
Yes, for sure. Yeah.
All right, we'll add that to the chat. So, but it's really easy, right? It's you had it up on the last slide, but yes. It's Nina B at SCCWRP.org.
Exactly.
Org. Okay.
Mm-hmm. Yes.
Okay.
Yeah, always welcome the questions from the next generation, of course.
Yeah. Okay, a couple more questions. Debbie wants to know, what are the species that are native to the area in Long Beach in the Colorado Lagoon?
That I presume that will be impacted by ocean acidification?
I assume so, yeah. That's the only thing that she wrote. But yeah, I'm assuming, yes. I guess maybe are there species, local species in the Colorado Lagoon that could be impacted?
Yeah, I don't know specifically Colorado Lagoon, but I can imagine that a lot of organisms is common between the estuaries and the lagoons. And mostly we're talking about, bivalve species, so mollusks, oysters, stuff like this that can be impacted negatively. There's a lot of crabs, green crabs, so variety of crabs that lives here. Lobsters is a good example that maybe ventures between the coastal and the lagoon estuary, and those are also the organisms can be more in the larval stage, more impacted. Let's see. Lagoons are actually very specific habitats and the estuaries, because they have a little bit of a different dynamics of a carbon compared to the coastal and the open ocean, right? Because of this really intense photosynthesis and the respiration processes between day and night, they see huge swings in CO2. So usually the organisms that are inhabiting the lagoons and the more estuary type habitats, they might have some more adaptation potential compared to the species that have never seen this kind of rates of changes in their natural habitat before. But I would say think of a variety of different calcifiers, especially early life stages, that will probably be the one that are not so good at tolerating more acidic conditions.
Okay. Next question is, Marie wants to know if she understood you correctly. Deep water species are more affected by pollution than top water species?
And the pollution, that means like toxicant, or does it mean lack of oxygen, or? I'm
guessing maybe she means like by the nutrients and affected by ocean acidification more so.
Okay. Yeah, so when the nutrients actually come to the water, they are usually retained in the upper 30 to 40 meters. They are usually used in that upper water column, and they don't go deeper. So species are not directly impacted by nutrients. They are impacted by nutrients being decomposed. That causes more CO2 in that upper water column. When they come into this big plume of CO2, then they can start getting affected. But it's not going to be on the bottom. It's going to be in the upper surface waters.
Okay. Evie also wants to know, how does methane affect ocean acidification?
Well, that's an interesting question. And as a scientific community, we really don't have much consensus about this, because the studies are really lacking how CO2 contributes to that, right? We know that the release of the methane can have into the atmosphere can have very negative effects, because it's such a potent gas. but in the water, to what extent it can acidify the oceans. I mean, the equation would suggest that it can have local impacts, but we don't have that understanding yet, to what extent it can impact the community. It's interesting, for example, that that organisms that do live close to these methane vents are usually much less susceptible to corrosive conditions, because they're very, very adapted to these conditions. So, and the vents are here and there, like along the West Coast, we have just a few examples of the ecosystems that are, you know, under the impact of the vents. So locally they could be important, but not on a larger scale. So if they're emitting the methane, it's going to be important impact on a very localized scale, nothing bigger. So, but it's an interesting phenomena, because if we study the responses of the organisms in there, we can understand the mechanisms, how some species are impacted and some are not. So the methane could be kind of comparable to the volcanic events, right? So for example, so submerged volcano are emitting these CO2 bubbles, and in this process, the local ecosystems are living in really acidic conditions. And that's kind of a insight of the future marine ecosystems, right? How they're gonna look like when the oceans are getting so much more acidic under future scenarios. So something that we can study in the nature itself, right? It's not experimental. And that's really important when we need to study not just the species, but also the communities. And so all the work that actually relates to these vents, CO2 vents, has shown a much lower diversity in the systems, where basically the coralline algae, for example, are gone and replaced by this fleshy algae, right, that can tolerate this CO2, but all the other classifiers are basically gone. So reduction in the community and really simplified food webs and systems. So maybe the methane can be used in this kind of way for the natural laboratories as well. Yes, that's the idea.
Okay, just a couple more. Let's see. Rex wants to know if you can recommend any good books or websites that may help the general public or novices learning about climate change and the effects that are happening either specifically in Southern California or worldwide.
I mean, there is a lot of information on the net, and I would almost like books are maybe more specific, but the internet has a lot of resources. The only thing that I want to maybe caution is like, how do you, in the amidst of all this information, how do you distill for something that is really critically or portrays something that is, scientifically credible versus just maybe some non-informed information, as we had the problem with CNN completely twisting our scientific story. So I would say read multiple sources, inform yourself, email me if you want about specific questions, but the more you are informed and the more different resources you're reading, and always check where is this resource coming from, the more you're gonna be able to understand how different maybe media are portraying this process of acidification, because you wouldn't believe it. The process of ocean acidification is also potentially differently explained depending who is telling the story. I mean, which political party is telling the story. So try to find unbiased sources in all this. But there's so much on the ocean acidification, on the new technology related to this. It's like an enormous source of information. So I'm sure you're going to need years to go through all this.
Okay. I have a question for myself, actually. You brought up that pteropods are the canary in the coal mine. And can you just elaborate a little bit on that, and specifically about pteropods? What's their life cycle? What's their lifespan? And where do they fit in the food web? And I guess, like, why should people care about pteropods? I care about pteropods, and I'm sure you do too, but why should the general public care about a teeny little snail out in the open ocean,
Mhm.
if they go away, you know, maybe nobody would even know that they're gone, but
Mhm.
what kind of impacts, like what's their place in the food web in the ocean?
Why should I care?
Yeah.
Yeah. So, I mean, pteropods are really so fascinating because one species can be found in all ocean basins, right? From the tropics to temperate to the polar oceans. That's so unusual because usually a species inhabits one particular habitat and then would not live in the tropics and in the polar conditions as well. So the species has this like, distribution everywhere. It's actually a really good use as an indicator if we want to compare how so for tropics, for example, is changing compared to the polar stuff. And then, so depending where they grow, they would have a different life cycle. So for example, in the tropics, it's very warm, species can grow very fast, right? And usually pteropods complete their life cycle in a matter of 3 to 5 months. But when you go to the polar regions and it's very cold, these organisms can live up to 2 or 3 years and they grow into this really gigantic organisms. So pteropods in the tropics are maybe 1 millimeter, tiny, but the pteropods in the polar regions are huge. So, and the bigger they are, the more flesh and the proteins they contain, and the more important they are for the food web, right? So salmon in the Gulf of Alaska eating bigger pteropods would definitely have much more energy from the pteropods than, for example, fish eating pteropods in the tropics, for example, right? And it's important to know that in the polar environment, pteropods are really important part of the zooplankton community. So they are very highly abundant, and all the fish in that super productive areas are actually feeding on them, right? So when you go to the local store and you say, hey, you know, salmon, widely caught salmon, right? What is that salmon eating? It's eating pteropods because that's in part of the season, that's the only food that they can get, right? Because everything else disappears already or transitions into maybe, so for example, salmon also eats crabs, right? But the crabs go from the pelagic to the benthic organism at the transition of the summer and winter period, and they are no longer available in the fall and winter, but fish still need resources to grow, and pteropods would be the source that they're feeding on. And if they didn't have pteropods, they wouldn't have energetic reserves. They might be feeding on something else, but they might need to traverse much longer distances to get that food, right? And so we don't know the implications of this, but if that important food source goes away for the fish we are eating, and it doesn't matter if you're in Iowa or you know or California next to the oceans, you are always buying the fish that is coming from different regions, and there the pteropods, regardless how small or big, can be very important food source for these organisms, right? And so we need to understand that we are living in this, you know, era of global massive consumptions, and all the sources actually travel, you know, so much that we no longer can just be thinking, oh, what is happening in my local backyard, right? I have to be concerned what is happening somewhere else in the Arctic or so, because I am getting my food resources from there as well. So it's really important. But so this is where pteropods are important for me and why I should care immediately, right? If I want to raise an awareness as a food source for this fish. But when we think of pteropods maybe more as a scientific item, right? How they can help us understand what's going on in the ocean. It's so easy to track this shell dissolution and changes in their habitats, right? We just collect them, put them under the microscope, and we are like, oh, there's a lot of dissolution. What's going on in this habitat? So by understanding the pteropods, we can understand to what extent we have already changed the oceans, right? So, and we cannot do that for a lot of species, right? Because we are always financially limited. We also cannot, some organisms do not have shells that we could quickly see what's going on, right? So if we have some tool, and I'm talking of pteropods as a tool that help us understand these changes, then we are utilizing it to better understand our oceans, right? And so they are also not just the indicators of oceanification, but indicators of ocean health. If we understand what's going on with pteropods, we can potentially understand what's going on with the copepods, with the crabs, with many other species that we cannot do all the research on, but we can have an insight. Oh, if this species is compromised, it's very likely that I'm going to see a comparable effect on the other species. So that's a direct implication of how am I going to monitor for ocean acidification? How am I going to manage for this? Where am I? To what extent do I understand these changes in the ocean health and the ocean system? So it has this role that really brings us much better insights of what's going on in the oceans, and I don't have to do a lot of other stuff, right? By detecting what's going on with the pteropods.
Cool. Thanks, Nina. Yeah, I think that's a great way to end it with pointing out that everything's connected, and the network, the ocean's a big place, and everything's connected and networked. So we really want to thank you for your time. So thank you to our speaker, Dr. Nina Bednarsek. And thanks again to the Friends of Cabruman Aquarium and the City of Los Angeles for their support. We apologize if we couldn't get to everybody's questions, but I didn't want to go too long tonight. But once again, I want to remind you of our upcoming lectures. The next one is going to be on Friday, November 6th. It'll be Dr. Nicole Benuso from Cal State University Fullerton. And the next one after that will be on Friday, December 4th, Dr. Jay Smith from Cal Poly Pomona. More information can be found on Cabruman Aquarium's website, where you can get the link just like you did for this lecture. And this lecture has been recorded, and we will post it soon on our website. So you can watch it again, you can share it, and you can email Nina and ask her some more questions if you have any. So thank you again to everybody for joining us tonight. And thank you again to Nina. What a fantastic lecture. And thank you and good night.